It is a funny business being a scientist. I decided at the end that I was very inspired. When I was a kid, the person I really liked reading was Peter Medawar. I don't know whether you ever read his excellent essays and sometimes the review. The review of Teilhard de Chardin's book, for example, was a magnificent bit of deconstruction. And likewise, what is it? Arthur Koestler wrote a book called something like On Creativity or something like that, and Medawar absolutely launched into. I love a piece of invective, actually, really. And anyway, but he wrote a book or wrote an essay called The Art of the Soluble, which I think is very profoundly good advice for scientists. And he points out, and I think it's probably a biological perspective primarily, that the whole trick of being a good scientist is to have a good problem. And we can all have simple problems that we can easily solve, and you get very little credit for that, although it's good to be able to solve them. And we can all think of big problems, like what is the nature of consciousness, for example, which would be, I mean, maybe it isn't even really a problem, it's so big, it's not clear it does actually have a solution. So the trick is to choose the biggest and most important problem that you can, that you think you can solve in a reasonable time. And that sounds very straightforward and simple. In practice, however, I think it's not at all easy, because you never know at a particular moment whether the time is ripe to solve a particular problem or not. I mean, you might, how was I to know that gene cloning was going to succeed in my lifetime? And that opened up a huge realm of possibility which was unimaginably closed when I was first a student. I mean, it's hard to convey the revolution that's happened. When I started out as a graduate student, there was no Xerox machine in the department, because Xerox machines had been invented, but only if you were a large corporation could you afford a Xerox machine. So we didn't have copies of anything. I mean, you typed your thesis and you had carbon copies. The calculator, we did have a wonderful calculator in the lab. It was a mechanical calculator, and when it did a long division, it rang a bell at the end to tell you that it had finished. And the electronic calculator didn't exist. I think it's the first ones, again, came in towards the end of my time as a graduate student. I was going to say I didn't get any mentoring at all. I think you get help from your friends, I would say. I mean, the all along the line. So, I mean, for example, the first problem that I got involved with was the problem of how you coordinated the synthesis of the two chains of globin with that of heme. And that problem really came about as a result of going to my first ever scientific meeting and hearing a very famous scientist called Vernon Ingram, who was the man who solved the nature of the molecular lesion in sickle cell hemoglobin. He gave a talk about this question of coordination of heme and globin synthesis, which problem had been around for some time already, perhaps by then about 10 years, as a matter of fact. Anyway, he gave a talk, and when we got back to the lab and discussed it, the graduate students and myself, we realized that he had completely misinterpreted his data. And for some reason, I can't reconstruct this now, we were sort of arrogant enough to think that it was, we could do it better. So we did it better. And that was my PhD. But I also enlisted the help of a very able friend, Tony Hunter, who's gone on to great things subsequently. And Tony and I sort of basically collaborated in our PhD. And I think, it's one of those cases where it often happens, actually neither of us could have done it alone, and it was really cool. I had friends who were geneticists, and I had to teach genetics in Cambridge because, well, I'd say teach genetics. It was cell biology, but it had a genetic component, and I realized I was very bad at it. My mind just doesn't work backwards the way it has to, I think, if you're a good geneticist. And but I was well aware of their activities, and I had friends like Michael Ashburn, who was a classmate of mine, and he was a geneticist. And it was interesting, our sort of paths sometimes collided and sometimes diverged. One of the nice things about growing up scientifically in Cambridge, we were exposed to all kinds of different influences. I mean, it was a very exciting environment. I remember going, for example, up the road when Francis Crick first announced the, I think Sidney Brenner announced the sequence of the termination, the amber and the ochre codons. I mean, this was a amazing tour de force seminar. And then Francis Crick first revealed the famous way the wobble hypothesis and how the genetic code is set out. So I mean, these are things that live with you all your life. I mean, and so you're looking at the cream of the cream. You knew these guys were sort of, if not Nobel Prize winners, about to be Nobel Prize winners, and you saw them in action, and you realized, I mean, the bad thing was that they were so much above us that they were sort of unemulatable, or so it seemed to us at the time. The good news was that actually, we knew some things that they didn't, and that was clear. So, it made you realize they were actually human. They had feet of clay. And it also made you realize that, in one sense, science is terribly easy and simple. You just keep on putting one foot in front of the other foot. But you must make sure that the back foot is firmly planted before you move the next foot forward. Sometimes when you look at kids do experiments, I always wanted to know the result as soon as possible. So you always, usually it was an autoradiogram of a gel. So I always took, it wasn't really a long enough exposure, but very often you could see enough to figure out what had happened. So overnight you could be thinking about what to do next. Whereas other people, I've noticed, they just sort of put it down and say, well, I'm going home now, I'm going to forget about this. But I think, when I was really active, I was thinking, eating, breathing science, not exactly 24 hours a day, but close to it, and not quite 7 days a week, but close to it, actually. I mean, you're a really, really total immersion. Well, I think one of the important things about being a scientist is to know your own limitations, and I'm only too well aware of my limitations. As the only exam I ever failed in my life, apart from the driving test the first time, was an advanced O-level maths exam. It was sort of calculus and complex numbers, and I just couldn't do that. So I think it's really helpful to know what you're good at and what you're not good at, and then if you're sensible, you'll get help where your weaknesses are, and then, but in return, of course, help people who. I mean, I've never had any problem. I mean, I think I have a fairly generous nature. I mean, I like to help people if possible. And people today, I've noticed, the young are apt to be a little bit paranoid and say, you shouldn't help him because, he might steal your results. Well, it probably happened to me once or twice that people did steal results. They probably would have had the idea or stumbled across it anyway. But you usually get so much more back than you give, actually. I've always been curious about things. I've always wanted to understand things. It always bothers me when I sort of don't understand things. And I think, and I like the explanations to be simple and clear. So I mentioned earlier that we got interested in this problem of how heme controlled globin synthesis, and in I think 1975 or 1976, around about then, we actually solved the problem, and it came as a sort of blinding flash of light. We had one moment you don't know what the hell is going on, and everything looks very foggy, and the next minute suddenly whoosh, you know what the answer is. I mean, it's absolutely amazing and very thrilling, actually. I mean, that. Something in that case, it was a sort of problem that I've been wrestling with increasingly for almost 10 years, I suppose. And suddenly, from not knowing what was going on to knowing what was going on, it was great. But thereafter, you've got a problem. Now you've solved the problem. Now you don't have a problem anymore. So what do you do? Well, so I knew And actually, funnily enough, I knew it was deeply embedded somewhere in the recesses of my brain. The same meeting that had given me the PhD problem from Vernon Ingram, the inaugural talk was by this guy, Henry Borsuk, who had actually uncovered the coupling of heme and globin synthesis, actually iron and globin synthesis, and had also worked on sea urchin eggs and discovered that when they were fertilized, they started making more protein. So I wanted to go and see where I could get some sea urchins. Now, Cambridge, unfortunately, is a really bad place for sea urchins because it's about as far from the sea as it's possible to be in England. It was an incredible stroke of luck. So me and some friends organized an EMBO workshop on translational control, because that's what we were experts in. And just as a sort of odd joker, I invited the person who I thought was probably the world expert on the control of sea urchin protein synthesis to come and give us a talk to say, what was now known about this. And it turned out that when he arrived, I'd never met him before. His name was Tom Humphreys. He said, do you know if there's anywhere I can rent a bike in Cambridge? And at that time, believe it or not, now you could. In that time, you couldn't. So I said, no, I don't know. I don't think you can rent a bike. But why don't you borrow mine? Here's the key, And so he borrowed my bike, and we became friends as a result of that. And then it turned out that he was the director of this course in Woods Hole. And I said that how much I wanted to work on his problem. And he said, well, why don't you come and help teach in Woods Hole? What I didn't know then was that actually it's quite hard to persuade people to teach in Woods Hole. But to me, it was like a sort of, the best offer, the best present you could possibly have given me. So, eagerly I went off to Woods Hole. Now, of course, I was used to American labs, and they're all incredibly well funded and well equipped and stuff. But this wasn't the case with the embryology course in Woods Hole, because the course was not equipped to do biochemical or molecular biological experiments. You couldn't run a gel there even. All you did was you fertilized various phyla, and you watched the embryos develop for the first 2 or 3 days of their lives, and then you went on to the next phylum. So, but we had a terrific time, and I invited somebody who'd done a project with me as an undergraduate, a guy called Howie Jacobs, who is now a sort of punk Finn, but then was a nice little Jewish boy from North London, to come and be my assistant. And Howie got involved in a car crash in Greece and was, I think, put in jail for a day or two, and arrived very bedraggled. But his first words to me, you do realize, Tim, that I only eat kosher food. It was a very challenging kind of way. And I said, Howie, that's not a problem. So poor old Howie then, from then on afterwards, had to eat these ghastly kosher meals that came up with some food service from New York, all lukewarm, wrapped in silver paper. But it wasn't a problem anyway. So we spent most of our time dancing and eating pizza and stuff like that. It was a very enjoyable summer, scientifically totally unproductive, because you couldn't do what I wanted to do. Then the next summer, I can't remember what happened. The course changed, and I wasn't invited back. But then a year later, I'd actually been quite a good teacher, I think, in my funny way. And the next time I went back, I knew exactly what to expect, and said, if we don't have a Revco, I'm not coming, and stuff like that. And I brought a gel apparatus in my suitcase, and I brought lots of Eppendorf tubes and tips and pumps and pipetters and all the rest of it, and began actually over the next few years to set up a little lab, a home away from home, and bought lots of plastic boxes from the local hardware store so you could keep stuff in it to stop the sea air getting. That was a bit of a problem there. That it's quite a salty atmosphere in Woods Hole, and when in the winter the storms get up and a lot of sodium chloride comes through the window.
But it's got sea urchins.
But it's got sea urchins, and it had sea urchins, and so I was able to start working on sea urchins, and quickly found that they're it was great actually, because we had all sorts of technical difficulties to solve, which we gradually solved, so that by the time it came to the time of the sort of famous Nobel experiment, I knew exactly how to do the experiment, and things were very nicely laid out. And that meant that the very first result was extremely trustworthy, right from the word word go. In other words, I knew how to fertilize sea urchin eggs, I knew how to prepare the samples, I knew how to analyze the samples on a gel. We'd got all the stuff for running autoradiograms. So it was, it was a case of chance and not only the prepared mind, but the prepared lab. So one tends to sort of forget about all of that stuff. I was reminded of it many years later, where I was invited back as a sort of consultant to the physiology course in Woods Hole, and I wanted just to take some nice pictures of clam eggs in their early stages of development, and I discovered, how very difficult it is to do that if you don't have all this, these sort of silly little things that you just need to have to hand, and if you don't have them to hand, you can't do anything at all. So the experiment was an incredibly simple one, and actually it was designed to look at something completely different from what it actually revealed. And I think, this brings up a point which I think is terribly, terribly important, which is that for me, the aim of science is to discover new things. And the thing about new things and discovering them is that precisely they wouldn't be new if you knew they existed already. So it's only when you sort of turn over a stone and there's something, a creepy crawly underneath, that you've never seen before in your life. Politicians hate that, of course, because they think that science sort of put on a white coat, go into the lab,
cure cancer.
Cure cancer because that's what you set out to do. That's not how it works at all. It's the unexpected things that are the really great breakthroughs. So I did this very simple experiment and analyzed the results, and I saw this protein go away. And there was no doubt that it was going away, and it was very quickly obvious that it went away in time with the cell division cycle. And so the sort of conclusion, because I've been thinking about this stuff for probably, I went there first in 1977, we're now talking about 1982, so we're talking sort of 5 years of thinking about cell division, these rapid cell divisions that sea urchin eggs do after fertilization, and wondering what was driving them, and to know that if you inhibited protein synthesis, you blocked that division, and to see something go away. Well, if it goes away, then you've got to make it again from scratch for the next. So it wasn't. This wasn't rocket science. You didn't have to have a sort of PhD in quantum mechanics to suspect. Although, of course, it needed proving that, you were really here looking at the heart, the pendulum, if you like, of the cell cycle engine. And this was so unusual and peculiar that it's literally true that nobody had ever even suggested this as a hypothetical, a theoretical possibility. People had been worrying about what they called periodic proteins, activities that appeared. But they'd never in their wildest imaginings thought that it might actually literally appear and then disappear. You could turn it on and turn it off, but because, people didn't understand. People knew about enzymes that digested other enzymes. Your stomach and your guts are full of them. But if you let one of those loose inside a cell, you'd just turn to mush. There'd be nothing left. So that how could you possibly, the specificity, you could have most of the proteins totally stable and one protein unstable, and only unstable, as we later discovered, for 5 minutes in a cell cycle of half an hour. It's amazing. This protein is totally stable until you enter mitosis. Then it becomes totally unstable for 5 minutes, and then it's totally stabilized again. Because otherwise, so the protein just describes a sort of sawtooth oscillation like that, and it carries on doing it for as long as the cells are divided. And that was, it's funny, some things you sort of find out very easily by simple experiments. Other things take years and years and years. So equally simple questions have answers that are not nearly so simple. It's funny, and for this reason, I'm always very keen to know how people know things. And luckily, in our education, again, going back to undergraduate education in Cambridge, we were very much so taught by saying, how do you suppose they found that out? Or how would you investigate? This is what we know now. What are the next, what's the next step in the investigation? We were encouraged very much to be rigorous about what was the evidence in support of a particular statement and how you might push push the things forward. And it became very clear to my generation, at any rate, that our ideas were likely to be just as good as the biggest experts in the field. And I think that was I think that was an amazing gift, extraordinary privilege for us to have as kids. Would anti-CDK inhibitors, would anti-cyclin inhibitors ever lead to drug design? Now, my view on this, which I think is shared by my erstwhile boss, Paul Nurse, was that actually this is such a basic thing in life that it's very unlikely that inhibiting the engine of the cell cycle would actually lead to an anti-cancer drug, because it's not clear where the specificity would come from. I mean, okay, cancer cells divide, but so do the white cells in your body which fight disease, so are the lining of your gut. The fact of the matter is, that we don't really understand things very well, is the truth. And at the same time as saying that I don't see how it's going to work, I would also say, don't let me stop you doing the experiment, because there might be some angle which I don't understand, and maybe, in particular, I still don't really understand the role of what's known as the D-type cyclins in cell growth and cell division. And the drug companies got onto this very quickly, and and they're really good at inhibiting enzymes like protein kinases, and they're very good at finding ones which are really rather selective for particular subtypes. So let's see what they do. And just the other day, I heard that actually from my friend Charlie Swanton that actually there are promising advances. I mean, that there is a CDK inhibitor which is in trials and is showing signs of working. I think early ones probably weren't selective enough, or were directed against the wrong CDK or something, or had side effects. Which, I mean, it's quite possible, of course, that actually the thing isn't working via inhibiting CDKs, but some protein kinase, one of the 550 or so that different ones that humans have, that it's really working by inhibiting something that you don't even know about. But nevertheless, the important thing is to do the experiment and see. If you look at the history of inhibitors, people have been through phases of trying to develop inhibitors of protein-protein interactions as being incredibly useful. You could inhibit a particular transcription factor, let's say. And some very talented, hard-working people have spent years of their lives trying to do that with very, very limited success. I mean, most people would tell you it's impossible. Fortunately, every generation somebody comes along who is either naive or over-optimistic, and I'm sure that sooner or later somebody will find something. But in general, they're not very druggable targets. A phosphatase, however, ought to be. I mean, because it's a small, it has a catalytic cleft and catalytic residues, and the example of ocadaic acid and tortamycin and one or two other things shows you that it should be possible to inhibit them. And I kind of wonder whether this the inhibitor that we discovered, the phosphorylatable, this is called endosulfine, or ARP19 or ARP16. It has a little core, and we stopped short of actually finding out how big the core has to be to inhibit. But I wouldn't mind betting that it was possible to make, synthetic homologs or analogs of that structure, which would be quite effective inhibitors. I mean, interesting to know, worthwhile exploring, I would have thought. But how do you know?